thank you to Natalie. I've known Natalie for over a decade now, as well as Jeff Farmer in diagnosis. I remember when Natalie came out to our clinic and set us up for FST, so she's been very helpful and supportive, getting my site, a very busy clinical trial site, set up and activated to be able to utilize FST as an endpoint, both in my inherited retinal disease clinic, as well as in a number of different clinical trials that use FST as an outcome. Again, just as a quick background, I'm a vitreoretinal surgeon and an ophthalmologist. I am an MD, and I'm not a PhD, and I'm not an engineer, and I'm not a technology person. So I luckily have Natalie and Jeff sitting here when the technology questions come up. So I'll just caveat this right up front that I'm a clinician, and I'm going to give you the clinical overview of why we're using FST as an outcome measure in clinical trials, and probably defer some of the technical questions to my diagnosis team here. I am a clinical investigator in a number of inherited retinal disease trials, as well as age-related macular degeneration trials. A number of them are gene therapy related. Some of them are oral drugs. Some of them are natural history trials. Foundation Fighting Blindness has been a really active collaborator. I've been with the Foundation Fighting Blindness. They first started me with a Career Development Award 20 years ago now. And now I am involved in a number of their natural history trials, which often use FST as an endpoint for inherited retinal disease. So I'm here today to talk a little bit about FST, or full field stimulus tests, for a couple of reasons. I feel very passionate about being able to use this outcome measure for clinical trials, and hopefully potentially at some point in the future be able to use this as an FDA approvable endpoint, because I personally believe that FST is an excellent quantitative measure that allows us to be able to measure an element of visual function quantitatively, accurately, reliably, and it's for a broad population of patients. So FST, again, quantified, it is patient response based. I don't like when people say, oh, it's subjective. Everything in life is subjective. Our most approved FDA endpoint, visual acuity, is subjective. So patient response. So patient response, absolutely yes, it is patient response based. But I think the important point here is it's quantifiable. What does FST do? Well, it measures the retinal sensitivity, the area of best retinal sensitivity across an entire visual field by testing for the lowest luminance flash of light that can elicit a visual sensation that is perceived by a subject. You can see here a nice little image of the color dome, the diagnosed machine. So this is the color dome, the patient's face is up toward the machine. And this is the button box with the red and the green button. So it is a patient response driven test. They are holding a button box and given explicit directions on what to do. And then this algorithm of these light flashes are elicited, and patients are provided instructions on responding if they see a flash of light. So that test approach, again, there's varying luminance of lights that are prepared, and they're paired with an audible beep. For each beep, the subject reports if the stimulus was seen or not using a 2-button box response box. And based on the response, we get this difference of luminance presented according to an algorithm until the metrics are met to end the test and report a threshold. So here's an example of a threshold that's elicited from this, the data output that we get from an FST. So the threshold is defined as the luminance the patient can see 50% of the time. You can see here when there's no responses, or no, I'm not seeing. And then you can see yes, I'm seeing. And so that curve in between, we get that 50% threshold, this Weibull probability function. And that is our threshold for the patient's light stimulus FST response. Probably very importantly, especially if you're interested in inherited retinal disease and interested in retinitis pigmentosa and a variety of other diseases where we're interested in broad versus cone-based sensitivity response, the FST is one of the very few outcome metrics that we have available to us today that can help accurately differentiate between a rod response versus a cone response. Why, how and why does that happen? Well, we can test using a blue light, and we can test using a red light, and we get different responses if patients have separate populations of rods and cones that are fired differently. We all know that rods are more sensitive to blue light than red, and so it leads to this notable difference in the threshold for those colored stimuli. And so you see the first metric here is the blue light rod response, which is at a lower threshold, and then a different population with the red light, 22 decibel difference in this particular patient of what the red light elicits. And so we can understand that the rods and the cones are different and both exist in this particular patient being tested. I like this figure because this gives you just a little bit of an overview of the paradigm here for who is FST useful for. And the answer for, in my opinion, is that FST is actually useful to a very broad population of patients, meaning both good vision, intermediate vision, and very, very low vision. You can see other types of metrics, so maybe a standard ERG or flash BEP. We need to have intact cells. Pupillometry can range over a wider range, but pupillometry is, although an objective test, doesn't really have any direct correlation necessarily with visual function. FDA has not endorsed pupillometry as an approvable outcome, and a relatively complicated algorithm and video required for pupillometry. Electricity evoked response, being able to electrically stimulate and bypass the photoreceptor system for a very visually impaired system without photoreceptors to find out, are there any intact optic nerve brain connections? Would it be reasonable to try to replace photoreceptors in an extremely low vision patient? And so that's for patients at the far end of the extinguished retina extreme. And visual fields, of course, we need to have a patient. So that's microperimetry, static perimetry, things we often roll out, Humphrey visual field, octopus visual field. We've all sort of heard of visual field. The problem with visual fields is that you have to be able to have relatively good vision and relatively good fixation, most importantly, fixation. So when I send my patients back to my clinical coordinator to get a visual field, if I get that patient sent back to me with poor fixation, there are definitely many patients with a level of visual acuity or nystagmus or poor fixation who just simply cannot reliably perform either octopus visual field, static perimetry, or microperimetry. So really important point here, for most visual fields, you have to have some level of accurate fixation. This is not the case for FST. So that is a huge take-home point of FST. You do not need to have good fixation to be able to perform FST accurately, to get a good algorithm, to get a good threshold. So FST can elicit correct responses and thresholds for patients with extremely low vision and poor fixation. Very, very helpful for a number of different inherited retinal diseases. I really like this slide because I'm a timeline person. I like to think about the landscape changes. So FST prototype development back in 2005, 2006. I was graduating from med school. Diagnosis FST commercially available in 2009. And then the voretigene parvavac, Luxturna, phase 1 clinical trial and follow-on studies, which were using FST. That's for LCA2 or RPE65 mediated retinal degeneration. Later in 2017, gets an FDA approval. That was Spark Therapeutics, a company that got the first historic landmark, the first retinal gene therapy for an inherited retinal disease we all remember was 2017, December 2017. And that did not use FST as its primary endpoint, importantly used multi-luminance mobility testing, the maze. You all remember the Luxturna videos of the kids going through the maze and doing a lot better once they've had gene therapy. But their probably most important secondary outcome, which showed correlation, which I'll show in 1 second, and really helped actually get that FDA approval, in my opinion, was FST. And again, FST was very useful in that population. A few of those patients could also do visual field. Some could not accurately because of poor fixation. RP65 is a relatively severe retinal degeneration with potential for poor fixation and low vision. And so then we see EMA approval for voretigene parvavac in 2018 and the rollout commercially. And so now we're in the commercial era of voretigene parvavac being approved in multiple places. Still being treated bilateral, sequential, subretinal gene therapy for RP65. Importantly, in the US, and I don't know if this is still applicable today, this many years out, but some of the payers were actually requiring FST as the rebate-related outcome, meaning that patients after the payers wanted to know, did I pay for something and did it work? And if not, I want my money back. And I'm sort of paraphrasing. But so they would follow FST after in patients who were treated with voretigene parvavac to ensure that there was still that FST improvement. Why did they pick FST? Because it was an incredibly important endpoint that showed really important 20, 30 decibel improvements in those patients. It's easy to roll out. It's a small machine and easy software program and relatively easy to roll out to clinics. And the comparator would be the multi-luminance mobility test. A maze probably bigger than this blue room that we're sitting in right now, with a high ceiling with fancy lighting, calibration, 2 to 3 technicians, and a 6-hour test. There are countries where it's just simply not even allowed to roll out a clinical trial that uses the multi-luminance mobility test because they can't get rooms. Certain countries you just simply cannot get a room because of how big these cities are and how much space costs in medical facilities. So MLMT is great, but for instance, there are 2 sites in the US right now that currently have a multi-luminance mobility test, so its rollout commercially is challenging. And certainly payers can't require that to be followed, but FST they can because it can be rolled out. This just shows that there was a correlation of multi-luminance mobility testing and visual field in the phase 3 trial of retina for biallelic RP65 mediated IRD. In that trial, about 30 patients were treated bilateral subretinal injections in their phase 3. That was randomized 2 to 1. And then 1 year later, the deferred patients did have treatment. Their primary outcome was at year 1, like I said, multi-luminance mobility test. But that FST was extremely helpful as well, with 20 to 30 decibel improvements in the patients as a secondary outcome that showed efficacy. And importantly, there was a correlation. I think the number here was a 0.74 Pearson correlation coefficient. I'm not a statistician, but that is apparently a good correlation coefficient between MLMT and FST. So that was one of the studies that has been published showing a correlation between an accepted FDA-approved, approvable outcome, MLMT, and FST. And I think this is an important thing as we talk as key opinion leaders and different committees as we talk to the FDA and engage them, that there is definitely a correlation between an FDA-approved outcome, MLMT, and FST. The Lancet publication, this is a company that I've been an advisor for since its inception, 5 years ago, actually in 2020. That Athena Therapeutics was a biotech company. Well, it's much different when I talk into it, sorry. Athena Therapeutics is a company that was born in 2020. I think in the room here we have Laura Pardon. There she is right there. You can talk to her. She's high in the clinical development part of the company. And I'm an advisor. I'm the ophthalmology advisor in the company. And I've been a consultant for them for years. So this is a trial that used FST as a very important metric in another form of LCA, and this was LCA1, so Leber congenital amaurosis type 1, Gucci2D, 2 mutations in the Gucci2D gene, and this was published in the Lancet, I think a year ago. KCI Institute, a couple other sites were the key sites penned for this trial. So the design, there were 15 patients. They received 300 microliters of ATSN 101. This is a subretinal gene therapy. And they had a dose escalation part, cohort 1, 2, and 3, different doses, and then 6 in their dose expansion part B, adult and pediatrics. So 15 patients had their 12-month study period completed. And we are continuing to follow them for an additional 4 years for safety as per FDA guidance for any gene therapy trial. And the figures I'm going to show you now are the FST figures. So there was a mean change in dark adapted FST over time for patients receiving the high dose of ATSN 101. And these were in the 20 decibel range, similar to the, maybe slightly better than the retina and parvovac improvements that were seen in the LCA2 trial. So really profound, absolutely clinically significant, clinically meaningful, statistically significant, every word you want to use there, on improvements of FST in patients who were treated. You can see the red patients who were treated versus the blue patients who were untreated. There was also a dose difference. So the high dose was selected as the most efficacious dose. There was a dose response, and that was the dose that will be moved into the phase 3, which is currently being planned at this time. And this showed the correlations that were seen between FST and multi-luminance mobility tests. So MLMT was also studied. Actually, about halfway through the trial, we were able to get setups at sites for multi-luminance mobility test. Keep in mind, this was a phase 1/2, so we don't have our primary outcome was safety as for any phase 1/2. But the important outcome metrics that looked at efficacy were FST, and then this introduction of the MLMT halfway through the trial. So not all patients had MLMT, and there were missed visits because not all patients had had it at the beginning of the trial. But we did see MLMT improvement in the majority of patients who were able to have that MLMT assessment, and there was correlation seen between their FST response and their MLMT response. And there was a mean change in BCVA as well, so we can continue to look at all these correlations. of whether or not we see a correlation to BCVA and FST. I don't have the direct data on that to provide, but this is just a slide on BCVA. We actually did see a trend in improved BCVA in patients over time receiving the high dose of ATSM 101. So that's the red patients who were treated versus the untreated patients. So those slides give you a little overview of some of the trials that have looked at FST. There are hundreds. I could have gone through quite a few slides today of clinical trials using FST. A lot of them are inherited retinal dystrophy trials. We could go through a number of different companies interested in FST for pivotal outcomes as well, potentially in the future. But in conclusion, just so we can start some questions here, FST, as we talked about today, excellent quantitative, patient response based, but quantitative outcome measure for patients with all forms of vision, including those with low vision and poor fixation, which is actually very difficult with some of the other current day metrics, such as visual field, visual acuity, things like that. Rod versus cone differentiation is possible and allows us to give that decibel difference between the rod firing and the cone firing and see if we have different populations of cells viable in patients. There are a number of different metrics that we know correlate visual function wise or functional vision with FST right now. MLMT is probably the most important, and MLMT is an FDA approvable outcome as per the SPARK trial, and other companies are continuing to look at other mobility tests. And Foundation Fighting Blindness, a number of different committees, and some of them I've been quite involved with, have been working with the FDA. This is a work in progress, but working with the FDA and the regulators to discuss from our medical perspectives and from our patient perspectives, the utility of FST and these patient populations. And the hope is that in the future, we would be able to use FST as an approvable endpoint. I would like that very much as an investigator on behalf of my patients. I think some of the FDA requirements for visual function and functional vision, the thresholds are quite difficult and maybe not perfectly designed for all of the patient populations that need to have access to care. So at the end of the day, all of us are here because we like eyes and we like ophthalmology, and we want to have drugs and therapies FDA approved for our patients to be able to cure blinding diseases or halt blinding diseases. And so we need to work together as researchers, as scientists, as clinicians, with our regulators to discuss what works for our patients, what things are accurate. We want reliable data, statistically accurate data, but we also want things that work for our patient populations and show effect in a reliable way. So that's what we're working on, and it's a continued work in progress. But I think FST is a growing field, and we're happy to talk about any technical questions as well. But I have an FST in my clinic, and I can explain my perspective on it. I think it's relatively straightforward to use once you have it set up in your clinic, and I use it quite frequently in my non-clinical trial patients as well. So thank you for your attention today, and we can open up the floor to questions.
So the question was if you have manual control over the intensity of the flashes, if you can dial it down to dimmer or brighter. And the answer is that the algorithm is determined by the software, and the software does do that. If it determines, you've been getting a lot of yes, I can see that, it does go a bit dimmer. If you've been getting a lot of no, I can't see that, it does adjust until it meets the requirements for a threshold. And what we report as a threshold is that intensity of light, the patient has a 50-50 shot of seeing. The question was, can you use the FST to determine if there's sufficient broad population to see if they're a good candidate for gene therapy?
Great question. I think it absolutely a composite endpoint there to look at. It's not usually required in a protocol as an inclusion-exclusion criteria, so I'll say that. For example, for Spark Therapeutics, their label for Retina Parvovac is viable retinal cells per discretion of the investigator, which is very ambiguous. And most investigators look at the OCT, so we look at the structure. Is that the right thing to do? Probably not. So that's a great idea. It's not currently being done. I think it's an excellent idea. It probably will be utilized more and more as we realize the potential power of FST to answer that question up front. It's just not currently being done for inclusion-exclusion criteria.
So Scott asked about the status, I hope you don't mind. The FST has traditionally, if any of you have parallel this, is a 2-button button box. There's a picture on the presentation right now. And we have an updated design that plugs into PC which allows us to power a little motor inside so the subject gets a haptic feedback, a buzz, instead of an auditory beep cue. And this is great for patients who, because of hearing loss, because maybe they have ushers or something, are unable to hear the beep cue. It's also, we're finding, very useful for some patients who just have a hard time concentrating through the whole test. Patients who are easily distractible or tired. For whatever reason, having a little buzz in their hand seems to keep them more grounded. This is now in production. We do have it. The software can't understand it yet. Our currently released version of software does not interface with this new button box. Version 7 of our software will be released in the near future. We're hoping in the next couple of months. And that will be able to take full advantage of it. So the button box itself is available, but you probably can't use it for another couple of months is the answer.